Good morning, everyone. This is a lesson on the theory of localization. So open up your green books to page 17. So today we're going to run through a few activities, make sure that you understand localization, one of our first biological processes. So look at your definition you wrote on page 17. What is localization? It's the idea that every behavior is associated with a specific part of the brain or region. So we know that the amygdala is related to emotion, hippocampus is memory. So the idea is it only localized in that area? Is it only associated with the amygdala or the hippocampus? And so there's different debates on whether or not we have strict localization, no localization, or relative localization. And we'll be applying this to whether or not we think psychopathy is localized to specific parts of the brain. We talked about the other day nucleus accumbens and basal ganglia being with impulsivity, but is impulsivity only in one of those areas? And we've seen that it's in also the prefrontal cortex as well. So we will eventually get to the idea that really what psychologists have developed over time is the idea that we have relative localization. So one of the things you're going to be doing at the very end of this lesson is you're going to be writing your first SAQ for biology, and it's going to be on the question, explain one study related to localization of function in the brain. So we're going to be concentrating on one particular study, but we're going to bring in all kinds of other studies and ways that you can put in different sides of an argument for localization so that you can write an SAQ, but also eventually write a longer essay in ERQ. And so you'll have all of the information in order to do one or the other, which would be on paper one of the test. Okay, so one of the things that we can say about the debate, if you're talking about a central argument, when you start doing your intro, you're going to be wanting to talk about how over time there has been a debate over having strict localization, and then some people that oppose it, and then also the fact that we have relative with some newer experiments and newer technology that we've been able to study the brain. So it really started with strict localization. And we see this with the story of Tan. And Tan was a patient of Broca's, which you may recognize from the Broca's area that you learned about this lesson. And he was unable to speak completely. He could only say tan tan. We also have another patient called Along, and he could also not speak very clearly, but he can only say 5 words: yes, no, 3, always, and Lilo, which was kind of a mispronunciation of his own name. And then after his death, their autopsy revealed that Lalong had a lesion in the region of the lateral frontal lobe, which we know is the Broca's area, which is about speech production. So this is probably the strongest argument we have for strict localization, because we know that language production and comprehension are strictly localized to the left hand or the left hemisphere, and so we know that that's a very strong argument for strict localization. But this is a very early kind of idea about localization that we're going to expand upon. So look on 17 and 18 and look at what the Broca's and Wernicke's area is. And you can see it there on that diagram. That's the frontal lobe is where the Broca's area is, and then more towards the temporal lobe we have the Wernicke's area. So what is the Broca's area? It's the left lower frontal lobe, and again it's for speech production. And they also have problems producing sign language too. It's creating language in any way, which I thought was interesting too about that. I'll show you a video of this when I come back. It's 2 of the patients that have Broca's aphasia and then Wernicke's aphasia. Broca's aphasia is the ability to speak or find the words. And so a lot of times there's this stumbling over of words. They know it in their head what they want to say. It's just actually producing it with the mouth. And then if we look also on 17 and 18, what is the Wernicke's area? Again, left temporal lobe, and this is for comprehension of spoken but also written language. And aphasia is the inability to use the right word. So somebody might ask a question and they say, bat walked to street behind. And it seemingly to them makes a lot of sense, but it does not at all. So it would occur if there's brain damage and so forth. And also comprehending sign language as well. So it's not just for actual speech, it could be for reading and also for sign language. Somebody who gets in a car accident and hits their left side of their head on the glass window could have some brain damage and not be able to read, and they would have to use rehabilitation to learn how to read again. So again, I'll show you that video when I come back about that. So this discovery led to the lateralization of function. We talked about that, having that ability to do tasks from certain hemispheres and more control of the behavior in one hemisphere versus another, and that pons uses that ability for the right side to control the left side of the body and so forth. And this, we really do see more control of behavior on the left-hand side with both of these areas being in that hemisphere. The result is that left hemisphere is a cerebral hemisphere is responsible for language and speech, and damage on the right side would not affect your speech or language. And this will come really interesting when we look at the split brain. If you look on 18 and 19, your answers for this, these are a couple of them are actually going to be some studies we're going to go into more depth. But these are three studies that support strict localization. Chardé et al. Did a study on 9/11, and they found that activation of the 9/11 event, called a flashbulb memory. These are memories that are really imprinted emotionally because of something traumatic or something that happens. And the activation of the left side of the amygdala is responsible for that memory of that very imprinted emotional memory. And then Sachs and Kanweiser, they found that understanding that another person's belief is false, so another person's belief is false, is localized in the temporoparietal junction. And you can see it circled there, kind of between the parietal lobe and the temporal lobe. We have this belief that another person's ideas are false is really localized there. Very specific kind of thought process. McGuire et al., we will be looking at specifically with neuroplasticity, and they found that spatial memory, which a taxi driver would use, is localized in the hippocampus. And when we have learning new things versus old things, can actually shift the matter of the hippocampus from the front to the back of the hippocampus. So we'll be looking at that. But these are just other things that would be out there that would support strict localization, if that's the argument you would like to use. In addition, go ahead and turn to page 22 in your book. Turn to page 22. And here's some additional information that could also be used to support localization. Memory is always an interesting one, and I have one that also talks about relativity of localization. But this study was done by Wheeler et al., and they used an MRI. So again, they're looking for the function or the structure, of the brain. And so they were looking at rich memories involved in more sensory information. So they were looking at the combination of memories and perception and where that is in the brain. And so they told participants to study the names of common objects, and they wanted them to study the objects, so that's your memory, and they either paired the word with a picture or a sound. So for instance, if they were looking at the word dog, they would associate dog with either the picture or dog where the person could hear a sound. So those are those two conditions in that independent measures experimental design. And they wanted to see how many words the participants could remember. So they used the MRI to measure the brain activity. And then the results showed that they activated a part of the brain involved in sensation, and the visual cortex was actually activated when they were shown the picture, and then the auditory when they were shown the sound, which is exactly what you would think would happen. is if I'm being shown something, my occipital or visual cortex is going to be activated in my auditory if it's sound. But memories are a lot more complex and not just sensations, but also thoughts and emotions that are related to those memories. So we know that this is an example of where we could say that it has localization, but then it's more. In addition, on page 22, on the bottom there, you'll see this is actually where we recognize faces. And we do know that faces, when we do recognize them, they are on the right-hand side, but there are a couple of areas on both lobes that actually are face perception for survival, and the recognition of those faces can be there. So there's a debate whether the use of our faces are based on survival, or if it's just a visual area, that's just visual like every other thing. We will see with the split brain patients that the right hand side of the brain is actually very sensitive to faces and just the recognition of them, that they are a face, like visually, the structure of them. But we have this debate out there, this pituofacial area that's part of the occipital lobe. And again, they don't know whether or not it's just a normal visual or actual perception for survival. So we have a couple of. Then we have, if you guys turn back to 19, your answers on page 19 of your homework, we have Lashley. And he opposed strict localization. And this is in the evolution of the idea of whether we do have localization or not. So Lashley discovered that memory trace, and memory trace would fight against the idea of localization. So again, he thought that memory is or it was thought that memory is localized or stored in specific parts of the brain, and that your thoughts are in your cerebral cortex, which we've talked. So he had some rats run a maze to get food, and then surgically remove parts of their cortex. So he was basically damaging their brain. So the idea was, if memory of the maze is localized, then removing that area, then it would be impossible for the mouse to or the rat to actually complete the maze, because that part where it was localized or remembered would be gone. And what he found was that the rats were still able to run the maze no matter what part of the cortex was removed. So again, then memories are not localized, but distributed through the brain. So memory is not even distributed in the cortex, as Lashley thought. It's somewhere else in the brain. And again, we're going to use rats in this instance because we're not going to surgically remove parts of the cortex of a human. And again, our animal lovers who would not agree with that, it's the cost benefit of knowing that in terms of how it would benefit humans, and the idea of using a rat because of that. So out of this came the ideas of principle of mass action, which means that the percentage of the brain damage is proportional to the percentage of function loss. So when we do create, and back when they did lobotomies and took a huge portion out, the person was almost like a vegetable. But if you take little parts out, it's not going to be as bad for the brain in terms of percentage of function that has the ability. And it's not in a specific location, but the percentage of the cortex damaged. So he was saying, I took part of the cortex out, but if I took more out, then they would be able to do less. And it's not really the specific area, again, supporting the idea of distributed function. Equipotentiality is the ability of one part of the cortex to take over function when something else has been damaged. So this is interesting because it's related to, if you get brain damage, can your brain still function? And the idea is yes, it may, after the initial damage or to the brain, like a car accident or something like that, there's going to be some time for, slowing down of the brain, and then it will quickly pick up as the brain starts to reestablish itself and brain connections are reestablished because one isn't being used. The other side or other areas around the damage will start to pick up the function where the damaged one would have. But this takes a little bit of time, not a lot sometimes, it depends on how much damage. And that's related to what we're going to be talking about in a couple of class periods, which is neuroplasticity, the ability for the brain to form, reorganize connections, and so forth. So that's why it's so much more complex when we talk about local. being more relative. So this is the actual study that you are going to be using mostly, the most uh, detailed one that we have, especially for an ERQ where you have to write more of an essay. And it really builds on the idea of localization than distributed, which that's more relative, okay? So I would ask you guys to look at split brain patients, and you're going to be looking at a video in just a couple minutes about how split brain patient experiments work. And it's Berry and Gonzaga, and they they were supporting relative location. And again, when we look at these studies, we want to look at aim, method, procedure, results, conclusions, and we want to start looking at, okay, based on the conclusions, what's some strengths and weaknesses about the actual topic? How does this experiment really support or is good at supporting localization arguments? And then what are some weaknesses of it? Those are the things that are going to be placed in an ERQ. And so that's why it's important that we kind of look at strengths and weaknesses, either from an ethical standpoint, from a validity standpoint, from a design, sample standpoint. All those things are, we're going to be pulling in those Unit 1 activities to do that. So when we look at brain discovery, it really was in the 1940s where it seemed to have no effect on their intelligence or personality when they started doing these surgeries. And they were doing the surgeries, again, very much like HM, to disrupt the disorganized firing that was happening in the brain, where there was misfiring and there a lot of confusion in the brain. The brain just kind of shuts down and creates a seizure. And so they were trying to disrupt the firing from one hemisphere to another. So then after these patients who were already established, right, it's a case study in itself, but the experiment is an experiment. So we don't want to say it's a case study, but it's a very unique sample, of course, and so forth. And we're trying to apply something from a very unique sample to all humans, which is a limitation in itself. So they tested the split brain patients, and you can see how they do that. They're going to put objects, and they're going to have them grab objects. They're going to show one hemisphere, an object, but cover the eye of the other object. So at the bottom there where you see key, okay, normally in that, your brain's going to communicate, but in a split brain patient, it cannot communicate left to right. So if I cover my right eye and the left eye is seeing key, then I'm going to be able to say it's key. And one of the reasons why that is because the left is being processed by the right. It jumps over the corpus callosum, and then my Broca's area is able to communicate that it is a key. However, if I do the same thing with a split brain patient, you see the key in the left-hand side. It's being processed by the right hemisphere, like we talked about, but the corpus callosum can't communicate with the pons. There's no ability for it to jump over to the Broca's area, and so the person says, I don't know what I saw. But when you do the same thing and show the key over to the left-hand side, they will be able to either draw it or pick it up or whichever, because actually what the split brain patients are looking at is actually processed by the right. They just can't verbalize it because they aren't able to use that Broca's area along with the right-hand side. So when you're watching the video, okay, which we're going to pause this lesson and then play the video, watch all the different things, and also what the person does when they show books and faces and that whole new experiment that Gonzaga does in terms of looking at different ways and different objects. But you'll be amazed. Again, this is the idea that we have localization of the Broca's and Wernicke's area on the left, but amazingly, the brains are actually still, the hemispheres are still kind of working together, just in a very unique way. So go ahead and pause the lesson, and then we'll come back after you're done with the video. Okay, so we're back, and so we know that language, reading, speech function are primary in the left, and then recognizing faces, nonverbal emotions, and visual spatial tasks is in the right hemisphere. So puzzles, copying, writing, drawing maps, things like that are all those visual tasks. And it's really interesting when they go to grab things, draw things, He doesn't even know why he's doing certain things. He just knows that somehow the right hand side has recognized it, and he can't verbalize what he saw, but he's drawing out what he saw using his right hand side. So let's turn to the next part of your green workbook, and this is going to be way more information than you need. So I will post these slides up on Canvas so you can go back and copy them. So don't worry. I'm just going to kind of zip through aim, method, procedure so we can get to the other activities with localization. So again, their aim was to test the theory of lateralization. It's a special case of localization to look at a split brain patient. They want to see if the two hemispheres have any unique different functions. And they're looking at effects visually as the optic nerves from the left side are connected to the brain on the right side and vice versa. So again, their method, it's a quasi-experiment. They're not manipulating split brains. They're already existing, right? And it's in a laboratory, and they're using an independent measures design because they're going to do some tests with split brain patients and ones without. So the independent variable is whether the individual had split brain or not, and the dependent is their tasks and the performance on those visual and tactile, meaning the touch tests. The sample is a stratified sample, all epileptic patients, some with corpus callosum cut and some with not. That gives you your independent and your independent measures design. So those 11 were the split brain patients, and then some of them were not. And then the procedure is the participants sit in front of a board and look at a dot in the middle of it. The visual stimuli would be presented for 1/10 of a second, either to the left or right field of vision, okay? And then the variety of objects were placed behind the screens so that the participants could feel around with their hands. And they were also asked to draw, just like you saw in the video, or show or spell words and so forth. So the results were that when shown a picture in the left visual hemisphere, or the visual field, the right hemisphere, the patient saw nothing when they were asked to pick up an object of what they saw and didn't see. They felt around against the screen, and that was because a lot of them, obviously from the first picture we showed, couldn't jump over the corpus callosum from the right-hand hemisphere. But when they felt around on the screen, could pick up the correct objects, and patients couldn't explain why they picked up the spoon, but just like you saw in the video, that's exactly what they did. When an image was flashed in the left visual field, the right hemisphere, like the word pencil, they were able to pick up a pencil. So anything, they were still able to pick that up. The word heart was flashed on the screen so that he was presented in the left and art in the right. And patients said they saw art because that left-hand hemisphere was able to verbalize that, but they pointed with the left hand to the card with the he on it to put heart together. Some patients were able to spell simple words with their left hand using plastic letters. One was able to spell love, but wasn't able to say what words they spelt because they weren't able to verbalize it. Recreating a drawing from an image shown in both hemispheres, and then objects flashed on the screen. And there were some pictures of a nude woman. And if the picture of that nude woman was flashed to the left-hand side, the patient laughed and identified they were laughing because it was a nude woman. Again, left hemisphere, they're able to use that Broca's area to verbalize. If it was flashed to the right-hand side, it couldn't jump over to the Broca's area. And they saw nothing, but they would chuckle, and they weren't sure why they were chuckling because the two hemispheres weren't communicating. So super interesting. Sorry, one more time. That would help with a distributed and relative localization debate, also recognizing that language is dominant in that area in terms of speech production. Okay, and then when you want to talk about is its strengths and its weaknesses, so it's a quasi-experiment with no control group. As there's no manipulation by the experimenter, establishing cause and effect for taking these split brain patient results and then having them transferred to most humans, that would be argued it's more of an in-depth study and may not be able to apply to everyone, so that generalization might be a little bit lower. And then a positive, though, was that the procedure that was used had a mixture of different experiments and tasks that they had, either visual, with verbal things. And you can see that with Gonzaga, what he did with his patient on the video. And then also ethics. Barry did not need to manipulate anything. The ethics were there. He didn't create split brain patients and so forth. He used what was already there. Positive and negative was the no control group. However, it's important to note that in this study, a control group was not needed as the results for the task for people without a split corporal callosum were already known. So comparing one group that had it cut and not, there's no need for a control group. I guess that is your control group. And then negative for external validity, we just talked about that might be low because it's, split brain patients don't represent, all kind of normal people. They're kind of very unique. And positive internal validity was, however, even though this doesn't happen in real life, this selected presentation of stimuli increased the internal validity because Sperry measured the effects of the hemisphere disconnection directly. He was trying to directly do tasks that were testing one hemisphere versus another. And then maybe negative was the selection. The sample was only 11, so the sample is really low. So how can we generalize that to the larger population? So those are kinds of the things, and you wouldn't have to write all of these, but in a larger essay, you might want to take one or two of these and put those in your SAQ. You definitely don't have to put all of the conclusions, but you can put a general conclusion and a couple of specifics, and then maybe one evaluative statement, either a positive and a negative, would be great in an SAQ to get all those points. Okay, so we know that the brain distributes function. If you guys turn to page 23, so go ahead and turn to page 23. You will see some things about memory. Okay, what are different parts of the brain that talk about memory? So the cerebellum we talked about the other day is procedural. The amygdala encodes emotional qualities associated with memories. The frontal lobe is where you retrieve and organize information about autobiographical, like when your birthday is, events that happened to you, and so forth. The prefrontal cortex is problem solving and sequence events, not the events themselves, but what happened in terms of the sequence of events. Your medial temporal lobe encodes complex memories by forming links in multiple brain regions and transfers like explicit memories to long-term memory. And then, of course, we talked about the other day, the hippocampus is encodes. It consolidates memory from the cortex and then back up into long-term memory. So just looking at just distribution of memory in itself, maybe that's makes more sense to you. We don't have a study necessarily, except if you look back at the strict localization of the dog and the bark and the picture, you could use that. But if this is also another example of where we have distribution of function. Okay, we're going to take a second to read and discuss a localization modern study of language. So we're going to pause the video, and you are going to go to pages 24 to 25 in your green book, and you're going to read quietly on the whistled language. And after all your group members are finished with that, just have a short discussion with your group before we go back to the lesson and discuss, does the study support or negate the idea of localization and behavior? And does it support our conclusions that it's more relative than not? So we're going to pause the video or the lesson, and then you're going to read this silently and then discuss for a few minutes in your groups. Okay, we're coming back. Move on to looking at a video on localization. So turn to page 26 and 27. And this is the last thing we'll do before you guys start writing your SAQ. So you've got a lot of information about localization, but how would you organize an argument? So as you watch the video, you're going to fill out whatever you want from answering the questions about localization and behaviors of humans. And again, this is posted on Canvas as well if you want to go back and look at a specific part. But we're going to pause the lesson, and then you are going to watch and fill out page 26 to 27 on localization. Okay, moving right along, we are going to just review really quickly about localization and lateralization. Overall, localization is relative, and some functions are more localized than others. So points to consider when we talk about generally about localization is some functions are localized weakly, while several brain areas might be responsible for functions, but some areas are more dominant, like language on the left-hand side. Strict localization for sensory and motor functions are more higher cognitive functions such as memory, thinking, and learning would have strict localization. Some functions are widely distributed, like memory, and some components localized while other components of the same function are distributed. Most languages holistically functions while production of the speech, for instance, is localized in the left-hand area in the Broca's area. Localization is not static. Areas move around, and that's our idea of neuroplasticity. We'll learn neurons can perform a variety of functions with some limits, and there's still a lot to learn about the brain and us learning about. Going back to your SAQ, you can pause the lesson and stay on this, and then work at your table to try to complete your SAQ. If you have any questions, give me a holler and send me a message on Canvas. Otherwise, we'll see you next time.